In this video, I'll be explaining the most important things for step 1. This is from Pathoma chapters 1 through 3, the most high yield notes. All of these things you absolutely need to know to crush this test. I'll be explaining it in the simplest way that I know how. I'm going to go as rapidly as I possibly can, while being as in-depth as I can. If this is helpful, then like and subscribe. Let's start with the basics of inflammation. So the most basic principle of inflammation is going to be the response of edema and neutrophils. If you see these, it means acute inflammation. And this happens because of infection. Why? Because we're trying to eliminate the pathogen, or tissue necrosis, so we can eliminate the necrotic debris. We also need to know that this is a type of innate immunity. Boom, we're done. Next high yield thing about acute inflammation, TLRs, toll-like receptors. So these guys are present on the cells of the innate immune system. Think macrophages and dendritic cells. So they're activated by the PAMPs, and these are commonly shared by microbes. So CD14 is a co-receptor for the toll-like receptor 4, which is on macrophages, and it recognizes the lipopolysaccharide, which is a PAMP, on the outer membrane. Okay, so CD14 recognizes your lipopolysaccharide on the outer membrane. The TLR is going to activate, causing an upregulation of your NF-κB. That's going to be an increase of immune modulation. Okay, so the easiest way to think about toll-like receptors is these guys are going to be present on your macrophages, also dendritic cells. They're going to be activated by your PAMPs, and this is going to cause an increase of your NF-κB, which is going to cause an increase in the transcription of your immune mediators. Okay, that's how the TLRs work. There's also the arachidonic acid metabolites. So this is where phospholipase A2 will free the arachidonic acid, then COX is going to get activated, and then we're going to have our prostaglandins, so our vasodilation, our permeability, and we're going to have pain and fever. All right, so A2 frees arachidonic acids to cause this COX pathway to activate. And then COX is going to make your PG, so think PGI2, PGD2, PGE2. All right, so the PGE2, you think pain and fever, and all of these guys are going to mediate vasodilation and also increase vascular permeability. So those two things are super important for the downstream effects of your arachidonic acid metabolite. All right, so we got our toll-like receptors, we got our arachidonic acid metabolites. The thing you need to know about 5-lipoxygenase, again, this is all the most high yield stuff that you'll ever get from Pathoma. You just got to know this stuff, is that these guys produce your leukotrienes. Okay, so like our LTB4 that activates your neutrophils. And so like these substances right here, they're going to cause vasoconstriction. And so you could think of like bronchospasm, increased vascular permeability as well. Okay, so that's what the leukotrienes do, and those are activated by your 5-lipoxygenase. So these are just like pathways that you have to remember, your arachidonic acid metabolites. Okay, now let's talk about mast cells. So mast cells are like widely distributed throughout the connective tissue. These guys are activated by trauma, so we think of our C3A and our C5A. And these guys are involved in IgE cross-linking. Anytime you see mast cells, think of IgE cross-linking. So the intermediate here is definitely going to be histamine that causes vasodilation and also permeability. So vasodilation and also permeability. So if you have like tissue trauma, complement proteins C3A, C5A, or cross-linking, then you got to think of mast cells. So the immediate response, you're going to have a release of your preformed histamine granules. So bye-bye histamine. So the histamine is going to go and they're going to mediate vasodilation and also increase the permeability. A delayed response is going to involve your arachidonic acid, particularly these guys, your leukotrienes. All right, so that's some inflammation stuff. I know this is a lot to cover, but don't worry, you're doing great. Okay, the complement system. So you have your classical, we got our classical pathway, we have our alternative pathway, and we got our mannose pathway. So the classical, you get the C1, it's going to bind to your IgG and M, GM, classic company here. C1 binds to IgG and IgM, that's bound to the antigen. The alternative pathway, so microbial products are going to directly activate this one. They're just going to go whoop, straight in. Mannose, this is when the mannose binding lectin that's going to actually bind to the mannose on the microorganisms and activate complement. All of these pathways results in the production of what's called C3 convertase. And that in turn is going to produce your C5 convertase, and that's going to cause you to have C5b complexes, and that's going to form with your C6 and C9s to make an attack complex. So you can have a Big Mac from 6 to 9. So just thinking back to our C3A and our C5A, these are your anaphylotoxins. These guys trigger mast cell degranulation. That results in histamine-mediated vasodilation and increased vascular permeability like we talked about. And so your C5A, that's a chemotactic for neutrophils. It's like, hey guys, come here, neutrophils. And your C3B is what we think of for opsonization. So it's going to be an opsonin for your phagocytosis. And we talked about our MAC. That lyses your microbes by creating a hole in the cell membrane. So that's what the MAC does. Okay, so that's a lot of material. Let's talk about the Hodgman though. Hodgman factor. So this is made in the liver. It activates your clotting and your fibrinolysis in the complement system and the kinin system. So think of like bradykinin causing vasodilation, permeability, and pain. Okay, that's our Hodgman's factor. Activating your clotting stuff. Clotting and kinin, Hodgman. Cardinal signs you see with inflammation. So obviously you're gonna have redness and warmth, vasodilation, relaxation. So like your histamines, your prostaglandins, your bradykinin, this is all gonna cause, the increased blood flow, arterial vasodilation. Tumors can cause, some vascular leakage as well. So rubor, calor. If you speak Spanish, that kind of makes some sense here. We got our redness, we got our warmth, and then we got our dolor, right? That's some of our pain. That's a sensitization by our prostaglandins 2s. So that's going to be your bradykinin. Think of a guy named Brady who just hurts you. Your fever mediated by your IL-1, also your TNF. It's going to cause an increase of your COX, increase of PGE2 in the hypothalamus, and that's going to reset your thermostat. So that's how IL-1 and also TNF are going to work. So TNF, COX, PGE2 in the hypothalamus, and then you got your thermostat is going to be reset. So that's how that works. Okay, so pain, Brady hurts you. Fever, pyrogenesis by that PGE2, increasing the temperature caused by this cascade here. Now, neutrophils. Let's talk about the neutrophil arrival and their function. So first you have your margination. Okay, so that's vasodilation, slows the flow, and so cells are going to move to the periphery. So they go to the outside. Okay, now we have the rolling phase. That's going to be monitored by our P-selectins. So think like histamine, and our E-selectins induced by these guys right here that we talked about. So that's the rolling phase. And then we're going to have an adhesion phase. And this is mediated by your ICAM and your VCAM and also your integrins. So your integrins right here are going to be upregulated by your C5a and LTB4s. Okay, so ICAM, VCAM, integrin. So the adhesions, think of the CAMs here, the ICAM, VCAM. Also, one disorder that's important here is your LAD. That's going to be a CD18 defect. It's going to cause a delayed cord separation in your baby. That's going to be infectious, but without pus, and also neutrophilia is going to be present there. So that's adhesion. Then we have transmigration and chemotaxis. So that's in our post-capillary venules. And our attractants that we're going to be using here are going to be IL-8, C5a, LTB4, and just like bacterial products. And then we do phagocytosis. So there we have our opsonins. It's going to be like our IgG, C3b. These guys, if you remember from before, your opsonizing guys. So what happens here is the phagocyte is going to merge with the lysosome, and you get what's called a phagolysosome. Ooh, a powerful guy. So in like Chidiac-Higashi syndrome, you have the impaired fusion, and so you have the giant granules and albinism, and you can also get peripheral neuropathy. So that's Chidiac-Higashi autosomal recessive disease. That's when that fusion doesn't occur, so no phagolysosome. Okay, now the killing. How does the killing happen? Well, it's O2 dependent, right? So we have our different reactions. NADPH, so that oxidase is going to cause this. SOD is going to cause this, and our MPO causes this. So our different reactants and formations we need to know. So if you have like an MPO deficiency, for instance, that could be like infections with Candida, because we just can't make these products to kill things because it's all O2 dependent killing. All right, NADPH. If you have an oxidized defect, like in CGD, for instance, you can cause catalase positive infections, so like Staph aureus and like nocardia. Nocardia is going to persist because you don't have enough NADPH so that you can create these molecules which will kill nocardia and staph. All right, so just MPO, got to think of your Candida as well. So those are some problems you might see on test A. And the resolution, our neutrophils, these guys are going to apoptose about 24 hours after the stimulus is going to end, up to 2 to 3 days, it says in Pathoma. Okay, what do macrophages do? So these guys, their arrival is going to be 2 to 3 days after the injury. Marginate, roll, adhere, transmigrate. You think of this as like the superheroes rolling onto the scene. They have to do their little action-packed entry. These guys are derived from your monocytes. So they're monocyte derivatives. These guys are going to destroy debris. You've got IL-10, TGF by the macrophage. You got IL-8 from the macrophage as well. So IL-10, TGF beta, important there. These guys are going to be involved in like your resolution here. You got IL-8 from the macrophage. That's going to be involved in your continued acute inflammatory response. Abscess associated with your fibrosis as well, and chronic inflammation. It's going to be associated with your CD4 and your helper T cells. So these antigen presentations are going to cause your chronic inflammation. So now let's talk about chronic. We talked a lot about acute, but here's the chronic. So this is going to be lymphocyte plasma delayed. So your chronic does have the lymphocytes. So lymphocytes are going to be here. Persistent infections is what you should think of. Like your viral, your fungal, even your parasites could be some chronic causes. Autoimmune form material and cancer. These are all going to be chronic problems of inflammation. So our T lymphocyte. So our TCR and our CD3, these guys are going to recognize your MHC molecule. So your CD4 is going to be a T cell of the MHC class 2. So the way you remember this is 2 times 4 equals 8, whereas 1 times your CD8 positive equals 8. So if you multiply these, the MHC molecule with the class type by the number associated with the CD is going to give you 8. So that's the way to remember that. So CD4 activation is going to be where you have your B7 on your APC is going to bind to your CD28, causing the IL secretions to happen. All right, so here are the important things to know about your CD4 helper. So once again, 4 times 2 is equal to 8. That's how you remember the class associations with the molecule. Okay, so TH1 is going to be your interferon gamma. So here you think like your macrophages, your IgG, and then your TH2 is going to be leading to formation of your IL-4. So there's like your IgE, and also IL-5, which is going to be like eosinophils and IgA. Remember the mnemonic hot T-bone steak for that. All right, now let's talk about our CD8 positive cytotoxic T cell activation. So remember 8, they're going to be activated by class 1 because 1 times 8 is equal to 8. So these guys are on all your platelets and nucleated cells. And these guys are going to participate in your killing through your perforin granzyme activation or through the fast L to fast, so the fast to fast L activation through your apoptosis pathway. So CD8s, they're going to kill through these two mechanisms, perforin and then activation of your fast L. All right, now B lymphocytes, how do these guys work? So these guys are produced in the bone marrow and they undergo immunoglobulin rearrangement. So that's where you have then the naive IgMs and IgDs. So these are going to be activated directly by this like antigen binding here, causing the maturation. And so here we have CD40 on our B cell, then we have our 40 ligand on our T cell. So this is going to help us undergo isotype switching. And so we're going to have a class switching and we're going to have plasma cell maturation. So that's how our B cells work, produced in the bone marrow, undergoing this rearrangement. So we have our naive IgM and IgD. Then our antigen comes in and binds and we get maturation. And then they can be activated so we cause isotype switching. Okay, granulomas are all over this exam. This is a subtype of chronic. There are two types to think about, caseating and non-caseating. The most high yield for caseating with central necrosis is going to be your TB and fungal infections. Non-caseating, think sarcoidosis, beryllium, Crohn's disease, cat scratch. So these guys, you have the aggregates of your epithelioid histiocytes in your giant cells. So non-caseating, you can think of no necrosis for non. And so here the formation is just your macrophage presents the antigen to your CD4. This then causes IL-12, and then you get your TH1 subtype. And then you get your interferon gamma, which then activates your macrophages and causes them to form into epithelioid histiocytes. And that's why you see the epithelioid histiocytes on these granulomas. So that's how granulomas are formed. So the macrophage presents the antigen to your CD4 cells. Then you get the IL-12, formation of TH1 subtype. TH1 can then cause the release of interferon gamma, interacting and activating the macrophages, causing these histiocytes to form, and you get your granulomas. All right, that was the entirety of the high yield from Pathoma. This would be like chapter 1.5, the latest Pathoma. These mechanisms, you just have to know them on test day, so this is a light review. If this was helpful, then consider liking and subscribing. We'll see you in the next video.